In this tutorial, we're going to look at some of the background that's needed to be understood before we talk about protein synthesis in great detail. So again, there's a few more terms to understand here, and we'll try and keep this fairly simple and short. Let's have a look at this diagram here and just see what it's showing. If you have a very close look, you can see that this is really a strand of DNA. Alright, so we can see the double helix down this end and down this end. In the middle it might look a little strange, but really it's the two strands that have been separated from each other temporarily. Alright, so the two strands of the DNA are temporarily separated because this is the process that's involved when messenger RNA is made in transcription, which is the beginning of protein synthesis. But I want to just use this diagram to highlight two terms that you need to understand. When a piece of messenger RNA is made from DNA in transcription, one question that arises is, well, which strand of the DNA is it made from? Well, it's only made from one strand or one side of the DNA. The strand or the side of the DNA that messenger RNA is constructed from or is based on is called the template strand of DNA. So this is the strand here from which messenger RNA is built up from, as you can see here. The other strand of the DNA is referred to as the non-template strand of DNA. The other thing is that you can see, of course, you know about the nucleotide sequence, or the base sequence as it's sometimes referred to. The base or nucleotide sequence is simply the sequence of bases along the length of the DNA. So ATG, ATT, CAT is the nucleotide sequence, or the base sequence along the DNA. This sequence, if it's a gene, is coded, and it's coded in what we call triplets. So 3 bases at a time, or 3 nucleotides in a row, constitute what's called a codon. So for example, here you may have AUG, which is a codon, AUU, which is a codon, CAU, which is a codon. And the codon usually refers to the sequence of bases on the messenger RNA. This code has been worked out as the genetic code. Let's have a look at this in a bit more detail. Now again, you've seen this before in Unit 3. Let's again have a very close look at it because it's fundamental to understanding how genetics works in Unit 4. First of all, this code is a universal code. So just as we said in another tutorial that DNA is universal, that is DNA is DNA is DNA, it doesn't matter what organism that it's in, the genetic code is universal. That is, whether you're in a human, a horse, a pine tree, or a fish, or an amoeba, the actual code, that is the triplets along the sequence of DNA, always code for the same amino acids. So it's a universal code. It can be applied to any organism. So the genetic code box is often seen like this, something like this. It usually technically refers to the bases on a messenger RNA molecule, although sometimes you will see a genetic code box based on the DNA, and you do have to be careful. But of course, if you see U's, which are uracil, you know that you're dealing with messenger RNA. The code is usually read off as a triplet. So U, U, U. You see that here, U, U, U is a triplet code, and codes or corresponds to the amino acid phenylalanine, as you can see here. U, U, G corresponds to the amino acid leucine, and so on. And if you have a look across the code, you can see that every one of the 20 or so amino acids is coded for by various triplet codons. You'll also notice, if you look closely, that some of the triplet codons code for the message stop, and this will become more obvious when we look at protein synthesis. But these triplet codons that code for stop actually stop the process of protein synthesis, which would tell a ribosome when to stop adding amino acids. There's also a start codon, and the start codon, although it's not called start, is always this one here, AUG, which corresponds to the amino acid methionine. So this is the start codon for any protein synthesis, AUG. The code is described as non-overlapping, and what that means is that triplet codons are read off in sequence. So if you had a series of bases that looked something like this, AAU, C, G, G, C, then the codons are discrete. That is, AAU as a codon, CGG as a codon. But for example, you can't have UCG as a codon. Alright, that would be overlapping two different codons, and the code is non-overlapping. So each codon is separate and discrete from the one after or before it, and this is never mixed up. It's never read in a different way. So it's universal, it's found in all organisms, it's non-overlapping, and it's also considered to be a redundant code. And this might be a rather strange name. If something is redundant, it means it can be done without. Alright, it's not always needed. What do they mean by that in the context of the genetic code? Let's have a look at something like glycine. So the amino acid glycine, like a number of other amino acids in this table, is actually coded for by 4 different triplet codes. So if GGU appeared, we'd get glycine. If GGC appeared in the sequence, we'd get glycine, GGA and GGG. All of those 4 triplets all code for glycine. So if, for example, something happened in the copying or the transcription of GGU, there was some kind of mistake in it, instead of the protein not being made, it's still possible that 3 other triplet codons could still make glycine. If a couple of them for some reason became dysfunctional or didn't work, then there's still 2 possibilities for making glycine. So what they mean by redundant code is that because there are sometimes there's more than one triplet that codes for an amino acid, then you can do away with some of the triplets in reality and still end up with glycine. And you can see this happens in a number of cases. There's arginine up here, has 4 different triplets that code for it. Tyrosine has 2, serine has 4 different codes, phenylalanine has 2, leucine has 2, leucine here has 4 more, isoleucine has 3, and so on. And that's what they mean by the redundant code. That is, you can do away with a number of triplets for whatever reason, and there'd still be other triplet codes that could still produce or lead to that amino acid being put into the sequence. Let's finish this tutorial just by very quickly looking at the human genome, and we'll look at this in more detail later in the course as well. But what has been discovered about the human genome, and remember the human genome refers to the entire collection of all the genes that make up a human being, so the genome of any organism is the total collection of all the genes that make up that organism. What's interesting about the human genome is that it's thought, or it's been discovered, that perhaps only 1 to maybe 1.5% of the genes code for proteins. So about 1 to 1.5% are coding genes, which is extraordinary. There's around about 3 billion base pairs in the human genome. BP refers to base pairs, of which 1 to 1.5% code for particular proteins. It doesn't take much mathematics to work out then that about 98.5 to 99% of our DNA is non-coding. It doesn't code for particular proteins, and it's now thought that much of this DNA may have a significant role to play in regulating the expression of the coding sections. So it may have a regulatory role in making the coding genes actually work.